Okay, finally, we're going to talk about personal diagnostics, and so the application of these techniques to the ability to predict and even fix diseases. So we talked in chapter 3, way back in chapter 3, about Mendelian diseases and some examples of autosomal recessive, autosomal dominant, X-linked recessive, kind of those types of diseases. And so, for dominant diseases, you know whether or not you're a carrier for the allele that causes that disease. But for autosomal recessive diseases, you might be a carrier for a disease, but you have no idea that you have it. And typically, the way these diseases show up is either through inbreeding among families, or just rare examples of crosses where two individuals are carriers for the same trait. And so, you right now have no idea if you're a carrier for most diseases, and you might get married and reach the point where you're ready to have kids, and that individual also will be a carrier for some unknown number of diseases. And so what you really want to know is, are you both carriers for the same disease? This is just a list of 4 autosomal recessive diseases, but there's really a large number of autosomal recessive diseases. And being a carrier is not something that's rare. So most of us are actually carriers for some sort of disease. And so this is why inbreeding, for example, is so bad. This is why having offspring with cousins or siblings leads to such novel and dramatically bad disease, is because all of us in general carry some sort of disease-causing alleles. They're just rare enough that we can always count on our partner to not having that same carrying that same gene. There's 25,000 different genes. But ideally, we would not want to have to go through this process randomly, where we just hope that 2 individuals are not carriers for the same disease. And so, what if we can do that? What if we wanted to look at all genes that are known to cause recessive genetic disorders and ask the question whether or not we are carriers for those? And so this is now possible. You can actually do this with a company. Council was the original company. They've now been bought by Myriad Genetics. But the idea behind this is that you simply collect blood or saliva or some sort of biological sample that you can extract DNA from. And so, this DNA can be sequenced by next generation technologies. So you could sequence the entire genome if you wanted. But usually for cost purposes, you want to restrict yourself to the genes that are actually known to cause disease. So you can capture these with probes. So these are probes that have a label that allow you to actually bind and grab them. And again, they hybridize to regions of the genome that you're interested in. So we've covered this idea to death. One of the very useful features of DNA is that you can melt it and then re-anneal it, and you can re-anneal it to like a foreign DNA source. So the concept behind this is simply that. So you design probes to capture the genes that you want to. And so you've recovered the exome DNA fragment. So you've recovered just the parts of the DNA that encode for genes, and you can further restrict it to whatever genes you want. So you really limit the amount of DNA that you're sequencing. And then you can just sequence this on the next generation platform. And so now we can simply compare individuals to see whether or not they're both carriers for a particular disease. And so this individual might have a mutation in the CFTR gene and the HTB gene. This mother might have a carrier for a OCA2 and a PAH gene, but there's no overlap. And so we know with certainty that the offspring, even if they inherit the defective phenotype from the parent that's a carrier, they're going to inherit the wild type allele from the other parent. And so there's no real chances, unless new mutation occurs, of this individual having one of the diseases caused by these 4 genes. But if both individuals are the carrier for the same gene, so this father has 3 different carriers, a carrier for 3 genes, and the mother is a carrier for 1, so they both have that same factor, HBB, so hemoglobin gene. And so that means that if they're both carriers, they're both carriers means that they're heterozygote. And so now they're both, so the offspring now has a 25% risk for sickle cell anemia. And so going in, are we incompatible at any of our genes? And does that mean that we have a very high probability, a 25% probability of having an offspring with a particular genetic disorder? So one of the things, what do you do about it? I mean, that's the very tough question. So even if you know that you have a 25% chance, then what are your options that you're left with? So one option is that you can selectively terminate a pregnancy that would result in an individual having this disease. So after reproduction has occurred, you can get samples of the fetal DNA, either during an amniocentesis, and also in Chapter 2 we discussed using DNA that's circulating in the maternal blood, and you can get a sample of the DNA in order to determine, is this individual a carrier, is this individual wild type, or did this individual inherit both copies of both defective copies of the gene. So you could either do this using biochemical analysis, karyotyping, or just DNA sequencing. More ideally would be that instead of having to go through the process where you selectively terminate a pregnancy, which obviously accompanies a huge number of ethical issues with even doing such a thing, is that you identify individuals that are not disease carrying before you implant it. So this is now possible due to advances in IVF, so in vitro fertilization. So this is where fertilization of an oocyte occurs in vitro, so outside of the womb. And so you actually can grow this fertilized egg before you implant it, such that the embryo has advanced to the 8 to 16 cell stage. So at this point, there's 8 to 16 cells that are within the embryo. And so again, you immobilize the cell. This part just is kind of holding the cell and immobilizing it. And then you can bring in a needle that is actually able to suck out one of the cells. So one of the 16 cells is actually removed from the embryo. And then, the embryo is still able to develop. So even though it has lost 1/16 of its cellular content, it's able to actually continue to reproduce as normal using the 15 cells. They're able to adapt somehow. And so we can use this as a sample, as a DNA sample, and now we can actually test for the genotype of this individual. So this cell right here is diploid. So it has both copies. It has the copy from the mother and the copy from the father. And so we can determine the genotype of the individual using this cell. So is this wild type? Is this a carrier? Or is this an individual that's going to have the disease? So that is the extent of Chapter 22. That's always one of my most fun chapters, I think, to teach, just because it's nice to get into the actual practical applications of a lot of these techniques. So hopefully you enjoyed that, and that is the end.